Okay, all right. Thank you, everybody, for now I'm going to actually talk twice as loud as I did when you did the sound test and blow the levels. But I am Danny Miller from the Puget Sound Mycological Society, and really happy to be here. I've spent, I kind of bit off more than I could chew by deciding to do a comprehensive DNA study of the Pacific Northwest mushrooms, where I want to see a sequence of every single mushroom that grows in the Pacific Northwest, and to find out by sequencing it if it's what we thought it was, or if it's something new or different, or if there's more than one species sort of hiding that looks like one, or there are two or three things that look different, but it turns out they're all the same thing. Kind of answer all those questions. And I was expecting, because of MycoMatch, the mushroom identification program that Martin's been talking about, they used to be called Matchmaker, that Ian and I are co-creators of, Ian's got a database that tries to keep track of all the macrofungi that have been found, and there are about 4,000 of them. And so that kind of thought that that would be the scope of what we were looking at, and that seemed doable in one lifetime. And now I have probably looked at more than 10,000 sequences, and still 1 in every 10 or 20 sequences coming back is a new species. The biodiversity is incredible out there. There's all kinds of things, both things that look the same that turns out there's several species of, and also what happens is we just finding new things as they're moving in. The climate is changing and we're getting lots of sort of warmer tropical weather mushrooms showing up in the temperate climate zones, and we're seeing things go away that we haven't seen in a long time, and new things are showing up all the time, and the diversity is just astonishing. So this is probably a multi-generational project now, but this is I'm going to talk about today some of the techniques that we use to try and figure out what mushroom you have once the DNA sequence comes back. Because a lot of people think, and I used to think, it's great, we're now able to sequence things, this is going to be fantastic, I've got the sequence that'll tell me what it is. And it doesn't really. If you blast, which means compare your DNA sequence, there's something called the gene bank, or gen bank as it's usually called, where everybody in the world puts sequences of absolutely everything. So people are going there when they're like finding new COVID variations, they're going to the gene bank to find out what's different. A mushroom, a plant, an animal, absolutely every sequence that everybody makes, they've all agreed they're going to upload their sequence onto this gen bank so that everybody in the world can easily go to the internet and see everybody's sequences. Great idea. Except they didn't really understand how to do it so it would be useful. For instance, it was never required at first to say where on the planet you found your organism. So it turns out that's important. You kind of want to know if you're matched with a mushroom, was it found in Tibet? Was it found in the North Pole? Was it found here? That's really important. So people are now typing that in, but there's no machine readable way. So you ask the computer to tell you what are all the sequences from this geographic area. It has no idea. You need like some extremely clever AI to try and figure out what that is. These are all things that are trying to be corrected, but and there's a whole lot of other things that are kind of missing from this data that people are wishing that they required people to enter when they put it in. So let's say that here is the BLAST program. Just go like BLAST DNA. If you Google that, you will get this website. And it gives you a box where you can put a DNA sequence in, or you can put the reference number if it's already loaded into the GenBank. You get this 8-digit character code back that uniquely identifies it. You can just type the code for it instead. And so let's say that I sequence this Cortinarius and I want to know what it is, and it's KJ920058. So I put in the sequence into the box, and if you want, you can type ACCGTT. You can type it all in, and you hit blast, and it will give you the 100 closest matches in only a few seconds. It's actually comparing probably to billions of sequences, and it already has the top 100 matches in just a couple of seconds. So here are all the names that people have put on their sequences. And most of them say Cortinarius violaceus. So we can kind of think that with all these matches that are anywhere up to 100% match here, 99.83% match, really, really high matches to Cortinarius violaceus. You might think that that what you have is Cortinarius violaceus, but it's often wrong. It turns out that most of the sequences in GenBank are misidentified. Some studies, one study once said that was warning people, hey, it could be up to 30% of them could be wrong. And maybe it's because I work with Cortinarius and Russula, which are notoriously bad, but I found that it's actually probably most sequences are misidentified because errors get propagated. The first person to put a sequence up there was the first. They didn't have DNA to compare to. They just took a guess and they named it. And the second person said, hey, mine matches theirs. I'll just use their name. And the third person said, I'll use the name these first two people had. And now 150 people have all put the wrong name on their sequences going up there. So you can't really trust it. You can see here that you've got mostly Cortinarius violaceus, but there's this other species here, Cortinarius Hirsinicus, I'm not quite sure how to pronounce that, is another possibility. So you can't just trust what you get. You want to get some proof to see that those things are actually identified. And what's really cool is you can click on them and it shows you what the differences are. So here we have, and I'll just sort of highlight what the differences are. These are all 100% matches. Here's one that mine is on top, the top row of each pair, and the sequence that it's comparing to from GenBank is on the bottom, and it's almost identical except for one difference here. And let's take a look at this one. This one's the same except for one difference. And we go on, oh, this one has 3 differences. So you can sort of see. Now, how close does it have to be in order to be the same species? How many differences are allowed? Well, that depends. The gene that you may have noticed that this sequence here is only 605 characters long. That is not the entire genome. Mushroom gene sequences are about 10 million characters long on average. Now, the humans, we have about 4 billion pairs, and mushrooms have about 10 million. So that's like 400 times bigger. And humans are arguably more complicated than fungi, although some people might argue the opposite. So we have much longer genomes, but still, to look at 10 million of these ACCTTs would be way too much work. So what we do is we just pick one gene, usually at a time, to sequence, and we take a look at that one gene. And in order to tell, and you pick a different gene depending on what you want. The first thing we're interested in is, what is it? Is it the same species as something else? So in order to answer the question, is this the same species as something else, you want to see every little difference that you can to try and pick that apart. If you have a gene that mutates like once every 10 million years and the same species has the exact same gene, you'll never know if it's the same or not. You want a region we call junk DNA, which is a part of the DNA that actually doesn't do anything. It's there, but your body never looks at it and reads it. It doesn't determine your hair color or right or left-handedness or anything about you. It's just letters that are there that your body never looks at. So if they happen to mutate, it doesn't matter. Nothing's going to happen. So every single time that, like sunspots or whatever it is that causes DNA to mutate, every time that there's a mutation in the DNA in this particular spot, it has no effect on the organism. It can still have children, and those children can grow up to have children of their own, and everything's fine. Now, if there was a mutation in, say, the gene that said how many hearts you're going to have, and it like switched to, say, 3, and it tried to fit, that's probably going to kill you. Certain DNA mutations are going to be really harmful because they're in a part of your DNA that's very important in order to determine who you are. And so a mutation might happen in that kind of gene, and then, but now you're going to die. You're never going to have kids. You're never going to pass that mutation on. That mutation will be as if it never happened. So maybe 1 out of every 10 mutations will not hurt the organism and allow it to still have kids, and you'll be able to see them. And so those kinds of genes might mutate 10 times slower than the junk regions that really don't matter. Does that make sense to everybody as to how the junk regions don't? So the ITS, or internal transcribed spacer, is the name of a gene that doesn't do anything. It belongs in fungi. Plants also have it. And so we sequence that, and it's only about 700 characters long, 600 to 700 characters long. You can look at it on a page, and you can compare and see what all the differences are. Really nice and easy peasy. So, and it turns out that just from looking at the DNA of thousands and thousands of species, about 0.5% change is usually enough to indicate it's a different species, on average. Sometimes much less. There are organisms that were in climate change that were forced to evolve to survive really quickly, where the DNA that matters to help them change mutated really fast, and the junk DNA that's just like once every 1,000 years, nothing changed. It didn't have time to change. So there are certainly cases where the parts of the DNA that are supposed to mutate slower have more changes, and you can find absolutely identical ITS regions in different species. And also, you might find that there was no pressure on this. It was living in a very stable climate area where there were no new threats to it, and it just went on for millions of years without needing to change. And you might see a whole lot of changes, up to 3% of the junk DNA might have changed over thousands of years, and it's just still the same species. It didn't really evolve at all. So there's a wide variation, and you have to kind of look at other things. DNA is not the magic bullet that we hoped it would be, but you look at it along with all of the things you've always looked at. You look under the microscope, you look at the morphology, and especially the ecology. It turns out that if you had to pick sort of one thing that might tell you the most about a mushroom, the ecology might even be more important than the DNA in some cases, because the junk DNA could have changed anywhere between 0 and 3%, and you don't know if it's a different species or not. But if it's if this one's growing on conifer wood and the other one was growing on deciduous wood, that's a pretty big difference and can mean quite a different ecology. Or if it's one is attached to wood and one is growing out of the ground mycorrhizally, those are huge differences that the ecology can show you. So you have to take a look at the whole picture and the DNA can be one more clue to tell you that whether or not you should consider these things two different species. So out of 700 characters or so, 0.5% is about 3.5 differences. So if I see 3 differences, on average, I'm thinking, okay, maybe this is the same thing. And if I start to see 4 differences or more, I start to question and look at other things to see if they might be different. So there's another database that people came up with called Unite to try and be a little better than GenBank. And it only deals with ITS sequences, just this one gene. So you can go here and you can type Cortinarius violaceus and say, tell me all about it. And you can set a threshold, which says, how different should the DNA be for me to consider it a different species? And I'm going to say 0.5%. I'm going to be rather conservative. And I'm going to say, take a look at all of the sequences of Cortinarius violaceus. And here's the results. Nope, that's not the result, because I didn't click on the green part before I clicked on go. There we go. Okay. So what this is saying is there's 10 possible genetic species that people have named Cortinarius violaceus, all within 0.5% of each other. So 67 sequences were all within 0.5% of each other that people called Cortinaris violaceus. So this one is a real good candidate for being the actual real thing. But however, there were 7 sequences that were all the same as each other, but they were much different than the other 67. So it's a very similar looking mushroom that may be a different species that people also thought was Cortinaris violaceus. So which one is it? And then there's a whole bunch of singletons that like probably aren't it. So what you want to know, and what's really important, it turns out, is geography. If you may have heard people say this is a European mushroom, and we used to call it by the European name, but now we have our own name. That's because people used to not really care about where it grew on the planet, but if they found a mushroom here that looked the same as a mushroom in a book written in the, in the 1800s in Portugal, and the mushroom looked the same, they said it's probably the same mushroom. This is back before they realized that ecology was so important. But we have completely different trees than in Europe, and we have different climate. Like it's sort of temperate in the same way, but certainly all the trees are different. So mushrooms that look the same have much more often than we realized turned out to be different species just because they don't have the same trees back as they did back in Europe, and they've evolved into different species to be able to grow under these trees. If you took this mushroom and you took it to Europe and you threw it under European conifers, it would not grow. It would not recognize it as a mycorrhizal host. So in order to find out what a mushroom really is, you have to go to the place that it was first described from and sequence it to find out what it is. So whenever somebody describes a mushroom, they pick one dried fruit body, say this is the type, it's called the type, and this is the official one with that name. So somewhere there is a dried little purple mushroom, a Cortinaris violaceus, that's designated the official one. And whatever it is, that's what Cortinaris violaceus is. Now the problem is in the 1700s, they described mushrooms by drawing them. So the official type of Cortinaris violaceus is a cartoon picture. And you can't sequence that and get anything out of it. So what you have to do is you have to go back to the part of Sweden where Fries was, where he found it. And so he'll say he found it on May 31st, 1821. And in this field in Sweden. So you basically say, what kind of weather was there in 1821? Oh, May 31st, it was kind of a late spring, so it wasn't really quite hot yet. It was kind of more like an average April temperature. So you wait until April of this year, so like the weather's going to be the same. You go to that field in Sweden and you see if the same kind of trees and plants are growing around. You wait for the purple mushroom to grow up, you grab it, and you say, this is probably what he found. And you'll never know for sure, but you just have to stop caring that much. You say, we'll never know, but from now on, we're going to assume that this one that I found under the same conditions he did, and I can sequence this and find out what its sequence is, and you call it the neotype, a new type that you found. So let's take a look and find out where Cortinaris violaceus is all about here. There's something called Index Fungorum, which tells you a whole bunch of things. Oh, and it wasn't Fries, it was Linnaeus. It was a long time ago. Originally Agaricus violaceus, because every gill mushroom was an Agaricus. So in 1753, Linnaeus found Cortinaris violaceus, and he found it in Europe. That's real specific. And so if you look here, in 1983, somebody designated this picture that was drawn in a book in the 1700s as the official one. And then they decided then somebody went to that area and they found a new one and they made a neotype. Here we go, neotype collected by Moser, but it wasn't code compliant. And anyway, a whole bunch of academics argued over what exactly should be the right mushroom, and they finally figured it out. But anyway, it is from Europe. So let's take a look at the 67 mushrooms that all have a very similar sequence that people thought was Cortinaris violaceus. Unite has a cool feature where it can put them all on a map. So here goes. Here we go. Here's all the 67 of them, and here are dots in the map where they were found. So it certainly is found all over Europe, and it's also found in the east, and it's also found out west here. So it probably is a worldwide species. It looks like we do have it here, and this could very well be it. So now let's go back and let's see the competing concept. Now there were 7 mushrooms that were kind of different, and maybe it was that one instead. Let's take a look. No, this one only grows in the east and west of North America and has not been found in Europe, so it can't be the real thing because the geography is too different. So what I do then is I take a sequence from Europe that seems to represent most of what you think. If you take one of these sequences, you can feel pretty confident that you have a sequence of the real thing because you've kind of done your homework and made sure that the geography matches. And as we see here, if we take a look at the comparison that I did at the beginning here, right down here, Cortinaris violaceus from type material. They did find a new one and they sequenced it, and this is the type. And ours is a 99.5% match to the type sequence. So my mushroom is probably definitely Cortinaris violaceus, the sequence. It has 3 differences, and as I said before, if up to 3 differences, I kind of feel comfortable saying it's the same thing. If there were 4, I would start to worry a little bit, or maybe I'd close my eyes and pretend I didn't see that 4th one. So within 3.5, and you've matched it to the type, I can be pretty confident that the sequence that I got of my mushroom is Cortinaris violaceus, and I can give it that name.
How do you account for there being 3 differences?
Well, as it's moved across and drifted across, they've been isolated populations that haven't interbred, so like some changes to the DNA have happened. And as I say, you can even find 3% differences. You might find 15 or 18 differences can happen sometimes, but the organism never really evolved any difference or is the same species. It's the same species for so many generations that so many random mutations have happened, and it's just not different. So, so we can feel pretty confident here that that we've got Cortinaris violaceus. So I want to show you when you compare DNA, it can be really interesting. I want to look at 0068. Here we go. Let's compare to this one. So here we have 3 differences, right? It says it's 3 differences. But sometimes differences aren't real. If it's not an exact match in the DNA, the computer's just doing a straightforward comparison. If it's not the exact same letter, it's going to say it's different. But it's not always different. For instance, this difference here is that there's a G in mine, and in the other mushroom, there's nothing there. It's missing. Sometimes a letter just falls away, and then the other 2 letters kind of close the gap and replace it. That isn't typically considered as big a deal as a letter mutating from one to the other. This little piece sort of dropping and falling away. So that's maybe like a half of a difference, let's say. Look at this. My mushroom has A, and their mushroom had R. What the heck is R? It's A, C, G, and T are the only 4 possibilities, right? So there's more than one copy of the DNA inside of the mushroom. Like we have a double helix where there's 2 copies of DNA inside of you, and those 2 copies can be different. And there's maybe 50 copies of ITS, and each of those has 2 copies. So there's more than one copy. And what R means is that sometimes it saw an A, and sometimes it saw a T. So maybe its mother had an A in that spot, and its father had a T, and now it has both of them. So an R means that A is one of the possibilities that could be there in that DNA. It might be a hybrid, it might be, but it just isn't a real difference. Because A really is in that position in, say, half of the genes that are found there. So if you ever see a letter that isn't A, C, G, or T, you can pretend that it was a match. And same thing here. W, I think it means it could be either a T or a C, but a T certainly is one of the things found in that second mushroom. So this is kind of a match. So what looked like 3 differences is really a half of a difference. So if I saw this, I would be really comfortable that they were the same sequence because they're practically identical. So you actually do have to look at each of the differences and say, is this real or not? Yes?
Are the copies exact, the DNA copies?
They are not. Like when they reproduce, they try to be exact and errors might happen, but basically the different copies are in different places and they're exact copies of its parents in those same places, if that makes any sense. Yeah. So there's one more cool thing that I wanted to show you, which is the 6695. And here we go. So this one. This difference here, up top. So the my mushroom didn't have anything there, and the other mushroom had an extra T. So there were 4 T's in a row in my mushroom, and there were 5 T's in a row in the other mushroom. Well, a really, really interesting fact is that computers have a hard time counting how many of the same letter are in a row. If the letter is different, if it's A, C, G, T, it's going to get it right. But if you say T, 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 computer goes, I was bored. How many was that? I'll just say 4. I'm going to persona, but they really do. They're really bad at knowing that. So if you see that one says 4 and one says 5, I can almost guarantee you that they were either 4 or 5, and one of the computers just got lazy and counted wrong. So this is not an actual difference. So you can see why I said I only wanted to sequence 600 characters. If you want to look at the whole genome that's 50 million wrong, you'd say, well, you'll get better results. Why don't you look at all 50 million? Because I'm looking at them all one by one, and that would just take forever. So if you want to look at each letter, you probably, I would be saying that I've sequenced and looked at my 5th mushroom in 10 years instead of saying I've looked at 10,000 mushrooms in 10 years because they're just so long. So that gives you an idea of kind of what you're doing. Now, so what do you do if you want to have something that's, put a reliable identification on your mushroom? What you want to do is you don't want to blast against the whole world's database where half of them are wrong. You want to curate your own private database of sequences that you trust and compare against that. So I've spent years cultivating a database of sequences that I trust the names of, and I'm going to only compare to that database, and so that I can trust what result I get. So on my DNA website, for every genus, I point you to a file that you can use, that you can blast against to figure out what they are. So we're going to try that. So here is, we're going to take, what happened to, oh, here we go, blast. We're going to take the mushroom that we sequenced, KJ920058, and we're going to blast it against, let's see, Cortinarius database. Here we go. So here is a database of 1,000 Cortinarius sequences or so that I've, one by one, tried to name correctly. And yes, it took a while. So there's a version of BLAST where it doesn't have one box. This is the version where you just put your sequence in and you say go, and what it compares it to is the entire world. So this is a version of BLAST where you can say click on align 2 or more sequences, and you put one sequence on top, and you just fill this box with however many sequences you want, and it's only going to compare them to the ones in that box. So here we go. It's still really, really fast, considering, you know, every time I'm like, oh, that took 5 seconds, I try to remind myself it compared against billions of things, and it's actually quite fast. So here we go. 100% match to something that I'm pretty sure is Cortinarius violaceus, and within 0.5% of the actual type sequence. So we know what it is. So that's what, if you were to be analyzing DNA sequences, the first thing that you would be doing is you would be comparing it against my database. There's a few other people. Stephen Russell has one for all the East Coast, and iNaturalist. You can download and export a whole bunch of sequences from that. So you compare to a database that whose names you trust, and if you get a 100% match or 99% match or better, you can feel that you have a match. So then the question is, what if, so that's great. If you match with a sequence of somebody you trust, you're pretty much done. But if you don't, what does that mean? Well, then you go and you blast it against the entire world, and you try and see if it matches anything there. And if it doesn't, and if it does match something else in the world, but not yours, you found something that I just haven't seen before. But you probably do have a sequence that is valid. Because if you get, sometimes you can get corrupt sequences. It happens a lot, where the sequence is for some reason garbage and it has a lot of mistakes in it. So you think you found something new and you're wasting your time because it was actually a bad sequence. So you want to know if that happened. So if you actually do match 100% to something somewhere in the world, there's no way that there's very little chance that your sequence is going to be bad, because to have random characters show up and happen to be 100% match, you should go to Vegas if that happens, because you're gonna, you should go to Vegas. So if you don't match anything ever found in the world, that happens a lot, because not a lot of sequencing has been happening. There's thousands and thousands of mushrooms, and really rare mushrooms that have never been sequenced. You probably did find something that no one sequenced before, but you also might be wasting your time because the sequence could be bad. So how do you know the difference? So let's take a look at this cool iNaturalist mushroom, 264039659. So somebody found this cool waxy cap, and we take its sequence and we blast it against my giant file of all the known waxy caps, and we see what happens. I'll close some of these so that this doesn't get unwieldy. 90% match is the best. 10% difference. Nope, this is not a mushroom that I've ever seen before. So now you take it and you say, has anybody in the world ever seen this mushroom before? And it'll compare it against the billions of sequences out there. Yep, no one's, 5% is the closest that anyone's ever seen. And pretty much the upper threshold of possibility of being the same is 3%. I've seen anywhere between 0 and 3% be the same, but that's clearly outside it. So this is either brand new or it is a bad sequence. And we would want to know which is which. So now we open up . Let's close these. Okay. So the sequence that you get is just a list of letters, A, C, G, T, but the sequence came from something called a chromatogram, if this is Sanger sequencing. This is what the results look like of a sequence. You have this gel plate, and it's got a whole bunch of colors on it, and the y-axis is how bright the color is, and the x-axis is just sort of a different spot on the gel plate. So this is what a sequence really comes back as. And what you do is you read off the letters by seeing what color you have. Red means T, blue means C, green means A. So you can read the sequence here. It's T, C, because it's blue, C, A, T. And that's how the file is generated. So by just looking at the string of numbers you're giving, there's no way of knowing if it's good or if it's bad. But if you go back to the original color graph, you can tell. So this is what a clean piece of DNA looks like. And if this is what the chromatogram looks like, where it's very clear at each point what color is, that's very clear. There's no mistakes. But there's some parts of it that aren't so clear. Look at all these extra colors in here. There's some second mushroom DNA got intertwined with it, and it's reading two colors at once. And the color of the wrong one might end up being taller than the color of the right one sometimes, and then it'll give you the wrong number. So when I look at, if I looked at my waxy cap sequence and it looked like this, I would say, whoa, I do not trust it. It could be bad, let's resequence it. And you're saving your time by embarrassing yourself describing a new species that didn't actually exist. But if I looked at the chromatogram and it was all like this, it was perfectly clean, I would know that I could trust it and I could feel confident telling people you found something brand new. Another interesting thing to note about the chromatogram is it kind of takes a while to warm up. Look how ugly the graph looks for the first 20 or 30 characters or so before it kind of settles out. And also at the other end, yeah, wow, that's extra ugly. So it kind of all falls apart right there. Yeah, here it starts to fall apart. So people that give you the sequence are supposed to know that and not give you any of the ends, just take the good middle part. But there might have left a little icky squiggly part at the end there. So when you're actually comparing DNA like we were here, if you see a difference right at the very end, in the first few characters or right in the very end here, you can be suspicious that that's just because it was a dirty part of the chromatogram. So differences at the very beginning or end of a sequence are often not real either. So, and one other cool thing, look at this. This first mushroom, it goes from CA to AC, and the second mushroom has 1, 2, 3, 4, 5, 6, 7, 8, 9 extra characters. AAAT, AAAAC. So what happened there is all 9 of those just fell apart, just dropped away, and then the two ends closed up. But that was one change. They didn't drop off one at a time. A piece of DNA 9 long fell off in one event. So that counts as one difference. So when you're counting how many differences are really there, it's not 40 like it looks like, it's 1. And if there was 1 here, I wouldn't count it. If there's the letter Y, that one doesn't count. There's a letter Y there, that one doesn't count. So it's not really 40, it's maybe more like 10 differences. So it really pays to look at them one at a time.
So if you get a dirty one like that, do you toss it, or like what do you do with it?
You are given sometimes a forward read and a backward read, which should be the same, just in the different directions, and you can compare them, and maybe one of the directions is clean and one of them isn't. But often you just throw it away and you try to sequence it again. Yeah. Now, so that's how typical sequencing works, Sanger sequencing, which is the way that it's been done for many, many years. Now, probably a lot of you have heard of Nanopore. I want to talk just a little bit about how Nanopore works. It's really cool because it can sequence like 1,000 things at a time for like $1 a piece. So you basically sequence 1,000 mushrooms for $1,000, and that basically averages to 1 a piece. If you want to sequence 1 mushroom, still costs $1,000. So you probably want to wait until you've collected more of them. Once you get to 500, you say $2. Hey, that's not bad. You might do that, but you certainly wouldn't want to do 1 at a time and pay $1,000 for it.
And how much was Sanger sequencing?
$30-$40 a piece or so. So to get $1 a piece is an incredible savings. And as I talked about before, you have to have primers and chemicals that work for a broad spectrum of mushrooms. When you're doing Sanger, you might have, like, the largest plates are about 96. You do like almost 100 at a time is the most you would ever do. So you can sort of be specific to what mushrooms are in that. But when people...
The Sanger sequencing still does those 1 at a time. You just have a plate of 96.
Yes, yeah. Yeah, and you still, and you have to do them one at a time. So what you do with Nanopore is pretty cool. So you have this giant tray that has like 1,000 wells in it, and you put a different mushroom in each well. And then you have a row chemical and a column chemical. So you take like chemical number 1 and you put it in all of the first columns. Then chemical number 2 and you put it in all the second columns. And then chemical 3 in the third. And then you take row chemical 1 and you put it in all of them across. So every spot will have a unique combination of chemicals. It'll have, if your tray is the 7th one across and the 5th one down, it has column 7's chemical and row 5's chemical in it. And you can uniquely identify it that way. So then you take them all and you put them all in one test tube. So you have all 1,000 of the different mushrooms in the same test tube. Whereas normally you had to keep them all separate. You had to have a separate test tube for each mushroom you wanted to have sequenced. And if you contaminated them, you would never be able to know which DNA you were looking for. There had to be only one kind of DNA in it. But now in Nanopore, you can get one tube that has all 1,000 of pieces of mushroom in it, of the DNA, and the DNA has got attached to it which row and which column it came from so that it can find it again. And then you buy this thing for $1,000 that plugs into the USB port, and you just pour it into this machine and it sequences it for you. You can do this at home. It's really, really cool. Yeah.
I think you just answered my question, but so the chemicals are attaching themselves to the DNA and
Yeah.
being read out that way.
Yeah, so like AAAA means row 1, AAAC would be row 2, AAG might be row 3. So it converts a row number and a column number into DNA, and it sticks it onto the DNA. So you have to remember to cut that off when you give someone the DNA, or else you'll really confuse them. What are all these extra letters at the beginning? But yeah, so yeah, it actually prepends it as the DNA. So
That machine you're using,
Yeah.
plug in, is that a mini PCR?
yeah.
Okay.
Yeah. So no, not the PCR is a separate step. You actually have to make billions of copies in a centrifuge and spin it around and make it. And you do that regardless of if you're going to use old-fashioned Sanger sequencing or if you're going to use Nanopore. The first step of like making the test tubes and spinning it around is the same for everything. It's just the second step that changes. Yeah, yeah. So then what you have is so when you pour the water in, there's all of these little tiny pores called nanopores, because they're like, nanometers long. And when the DNA just slides through one of the pores, it reads it as it goes through. So you have you have this like machine with like 1 billion tiny little holes in it that'll read the DNA if something goes through, and you just stir this liquid and the DNA just starts swimming through the holes randomly. So basically this fungal will be able to read 1 million DNA sequences, and then it's tired and it's dead and you have to buy a new one. So if, let's say you have 1,000 different mushrooms in the soup that you've made that you want to sequence at the same time, and you stir it up, you hope that on odds, each of the 1,000 mushrooms can go through and get its DNA read 1,000 times each, and 1,000 times 1,000 is 1,000,000, and then the thing is dead and it dies. But that's just on average. You might get really unlucky. You might get this like mushroom hog that just wants to go through 30,000 times, and this other one can't get through, and one of your mushrooms might just never happen to go through one of the holes and not get read. That could happen. But on average, you're going to get each of the 1,000 mushrooms will get read 1,000 times, and then you'll be able to see the sequences. So why do you want to have them read each 1,000 times? If you just read it once, that's enough. Why are you reading it 1,000 times? It's because Nanopore is very bad at what it does. It makes mistakes a lot. 1% of the time that it tries to, at least 1% or a few percent of the times that it tries to read what the letter is, it gets it wrong. So you can expect that the DNA sequence you get back will have 1, 2, or 3% errors in it. Now, I've just said that you have to know with certainty that you're 99.5% the same, or else you'll never be able to know it's the same species. You'll never know what species it is. 3% is way too many errors. It's useless. It's just too bad. So they came up with a clever way not to make the machine any better, to have it make all these mistakes, but to still be able to give you the right answer. And here's how it does it. There's this program called Eugene that lets you see this. Okay, so here is this particular mushroom got sequenced 494 times. So typically you might expect there to be a mushroom to get sequenced 1,000 times. And what they usually do is, because the software would be too slow, they usually just take the first 500 times that it's read and they show that to you. So here we have, so if we go across the top, each row is one of the sequences of that mushroom. So it's, and it goes over and over and over and over and over. So now they haven't been aligned. Let me just quickly align them. Because if one little letter fell off in one and it didn't in the other, they'll be off by one the entire way. You'll never be able to see them. So it has to put all those gaps back. So here we go. So this is from the top 1 through 494 sequences, and going across are what the sequences are. And look at, every once in a while there's a difference. These are all of the errors and the mistakes that the DNA sequencing does. It got it wrong. The sequence really was TTTG, and when it read it this time, it made 2 errors right in a row. It said, I think it's a C and I think it's a T, and it missed the G altogether. So these are just the mistakes that the machine made because it's not very good. Yes?
So why are the mistakes made? Is it that like the differences in its electrical charge?
I don't know. Like if you were to show me the molecule and ask me if it was an A or a C, like I might mistake it too. So I personify a lot. I'm not quite sure why, but yeah, but for some reason it's not that reliable.
Measuring electrostatic
Yeah.
charges?
I would make those mistakes too. Yeah. So here's a mistake. It didn't notice one of the G's that was really there. Here's a mistake. But here's the thing. When you have 500 copies of it, the mistakes are all going to be in different places. And you take a look at this, you can tell what it is with extreme confidence. Like, I feel really confident that I could tell you exactly what that sequence is with no possibility of error, because I've got 500 copies of it and it's very clear that that's the one that's wrong and all of these are the one that's right. So you can look at that and you can actually get the right answer. So that's why you're sequencing each thing 1,000 times. So you have all this redundancy so that you can figure out what it is. And that's kind of how Nanopore works. So it's really cool because you can sequence so many things at the same time and mix them all in the same liquid and not have to have 100 different test tubes, but just one big test tube in the end.
So Danny, just like one fungal is used for one sequencing run for that.
Yes, and then you throw it away. Yeah,
And that's all.
yeah. And then it's tired or broken. Yeah, it's not reusable. So yeah, so it can do a million sequences. So you can put 2,000 in there, and maybe you get 500 reads of each, and that's plenty, right? Maybe you could probably sequence 5,000 things at a time, and it still works. But the more species you put in there, the more likely that the errors won't be able to be corrected, and the more likely that some hog won't let the mushroom you cared about ever get read. So people tend to put 1,000 mushrooms at a time in there and get pretty good results. So let's say you do have a new mushroom. Now you have to name it. Now you have to figure out what it is. There are so many mushrooms out there that people don't know anything about. You can't really be confident that you found a mushroom new to science. You've likely found something that's in some obscure textbook somewhere that everybody's forgotten about. But for now, for our purposes, it doesn't matter. I basically give them all code numbers. Pacific PNW01, or like I'll call it hygroscopy PNW01, and the next new one I find I'll call hygroscopy PNW02. And yes, some of them may already have names, but that's okay. It just means we don't know what it is, and we know that we have to go back at this thing and look at it. So Mycomatch, which I'll quickly show right now, is, so here's all the guild mushrooms. In Mycomatch, you basically start with a category of mushrooms because they're all so different looking. You ask different questions about them, like how closely spaced are the gills are not a question that would apply to morels. So you sort of start by saying, what kind of mushroom do you have? And I'll say guild mushroom. And here it says 2,436 guild mushrooms known from the Pacific Northwest. So that's a lot. And here are all their names from A to Z. And you can basically click on one and you get a description of it. So this is a really cool resource because, like for instance, 1, 2, 3, here's all the agaricuses. They go down to here. These are all agaricus species that have been reported. I don't know about you, but I know there's like 5 or 10 agaricus that you might recognize. I think there's 40 or 50 of them. This is a great resource to take a look at all the ones you've never heard of and kind of eliminate them as possibilities for what you found. And if you want to take the time to do that, you can look through the literature and you can try to figure out what this new mushroom that you found was. But an important piece of work, probably the most important bottleneck that needs to be done, is all of the type mushrooms of every species have to be sequenced so we can compare to them. Right now, everything that I'm doing is kind of just guessing, but there's different people are, with various amounts of enthusiasm, starting to try and sequence all of the original types of every mushroom. So if every type had a sequence and you had a database of that, you would right away know if you found something or not. And we're trying to build that database. But the thing is, a lot of them are 100 years old and they're really hard to sequence. So the first thing you have to do is try your best, or send it to a specialist like Matt Gordon in Oregon, who's had success with 100-year-old mushrooms. And if you get a type sequence, great. And if you just can't, you're going to have to do what I described about take a plane to Sweden and walk around the moors where Linnaeus was in 1821 or whenever that was, and find the one that grows there and say this is probably what they had and sequence that one. So that's a very important project that's being done right now because that's the only way to get certainty in your answers, basically. So then when it's time to describe it, as I said, you don't want to rely just on the DNA. You want to look at the ecology and the microscopy and use that with the DNA to be confident whether or not this thing might be the same or it might really be different, and give it a name. And so that's kind of how what the process looks like. There's some more I can say, but it's been about an hour now, so I think I'll start taking questions and see kind of what you want to know more about. Does that make sense, sort of the process here? Does it feel like this is your life's calling? See me afterwards because we could really use the help. Or never gonna think about DNA again. That might be another logical reaction. Yeah. Any questions about this process so far? All right, let's see, what else can I, oh yeah, let me show you some trees. Like I can play with trees. What?
We just learned ecology in trees so we can figure out what the mushrooms are.
Yeah.
So these ones that you're showing right now, do they have a picture too? Like when you click on
Yeah, anything that has a star has a set of photos. So you can click on them and you can go through all the different photos of the species.
So you don't really use book if you use this.
Yeah, it's got a lot of data. So Ian he's the one who's entered, Ian Gibson from the South Vancouver Island Mycological Society started this in 1999, and he's the one who's typed in He has an extensive library, and he's typed in everything there is to know about every mushroom. And he's also written a notes section at the top, which kind of talks about it in his own words. That can really be a great summary, so you don't have to go through all the technical details. And at the bottom, he's also written a similar section where he talks about the ones that you might confuse it with. But the sort of bread and butter great thing about MycoMatch is the synoptic key, where you aren't asked questions that you don't know the answer to. Have you ever used a book key and it says, was it found on wood or on the ground? You're like, I found it on the table in the gymnasium. I don't know. So then it's like a choose your own adventure book, right? You know, go to page 1 if it was on wood, go to page 6. Now you're dog-earing both page 1 and page 6, so come back to page 6. Then it says, what color are the spores, or are the spores ornamented and you don't have a microscope with you? Like, well, now soon you've got like 16 pages dog-eared that you're looking at and you're just lost. So it's much better if you tell the computer what you do know and it doesn't ask you any questions that you don't know. So I'll give you an example here. So for instance, let's say I knew that the cap was red. It's got a whole bunch of characters, hundreds of them for each part of the mushroom. Here's for the cap. You can talk about the cap shape, the colors, the textures of the surface along the margin. For the flesh, you can talk about the texture and the colors of the flesh. For the gills, the gill attachment, how many gills there are, whether they're... Yeah, you can do as much or as little as you want. And you want to do just a few because you get yourself in trouble by really going crazy clicking a whole bunch of things. The stem shape and color, whether or not it had a veil and what kind of veil. All the odors it might have had. How did it taste? What color was the spore? What did you see under the microscope? Where did you find it? On the ground, attached to wood? Was there milk? If so, what color? Chemical tests, put KOH or iron sulfate or PDAB on it. What color did it turn? Or just scratch it and see what it turns. Anything you can imagine, you can say. Yes?
I have two questions. What can you share about the statistical methods that are used in this program, or these programs? And two, where can we find this specific program?
Yes, mycomatch.com is where you can find this. It works on PC or Macs. It is not an app for a phone, but we do have beta versions for both iPhone and Android. So contact me if you are interested in getting that. They only work for gilled mushrooms, not non-gilled yet. And they're just betas, but they're kind of fun to play with on the phone. So as far as the statistics go, it's only as good as the literature is. So sometimes the book never said what color it's going to change. It'll change extra colors. It never said all the possibilities of the shapes or all the range of sizes. So you're going to not find it often just because the literature didn't know everything. So the way that we try to compensate for that is you don't have to be 100% match. You can then go down to, if you don't find it, you can say, how about 90% match, 80% match, 70% match until you find it. And then I'll demonstrate that. Actually, it might make more sense. So I'm going to say here that I found a red capped mushroom. The choices went down from 2,300 to 142. So already really, really great way of limiting it. And you want to kind of think about what the distinctive features of the mushrooms are. Because if I were to say that the cap was some shade of brown, I will go from 2,300 possibilities to 2,100 possibilities. That really doesn't eliminate very much, as you can imagine. So you kind of want to be as specific as you can. So for instance, let's say it is red, and on the surface, there are warts on the surface. Oh, look at that. There's only 5 possibilities, and the very first one to come up is Amanita muscaria. So with something distinctive like that, it can really, really help you figure out what it is fast. Let's see what the other possibilities were. Amanita rubescens, yeah, that could be like that. A cystodermella, well, that's kind of interesting. And a russula. This russula was reported once with warts on the cap. So Now, let's say your mushroom wasn't there. You can say reduce by 10% and match again. So here's ones that, let's say you actually had the Prince mushroom here, and this is what you were looking for. Only 50% of the characters were an actual match. So you can actually say where is it? Show species characters. So it'll show in red the character that you insisted it have that it didn't know could be red. Let's say you found a red Prince because, I don't know, sometimes it might happen, and you put in that it was red. It shows the word red in red here to say that you said you wanted a red mushroom. I didn't know that mushroom could be red, and that's why the mushroom didn't show up in the list at first until you said relax the restrictions.
So is this an app that you pay for, or is it a?
No, it's free. It's on the auspices of the Key Council and SFIMS Society. So it's, yeah, it's.
So we use this here this weekend to help identify stuff that we have?
Yeah, I haven't had a chance yet, but I certainly do use it a lot when I'm really lost about something, because it also has like some of the chemical features that you can put in, and it's got some of the microscopic features too. So it can be really handy. Don't go crazy. Try like 3 characters at a time, and then look at the list. And then if you don't see it, maybe try a 4th or a 5th. The most common thing one of the really common things that happens that makes it hard to use is when people go crazy and put 6, 7, or 8, 9 things, and you're more likely then to hit upon a character that wasn't in the literature that we won't know about and prevent you from getting the match than if you just keep it to a few distinctive things at a time and work your way through it slowly. Yeah.
Is there any information or like a database on specific mushroom genes and what they might be responsible for?
Not that I know of. I don't think that there is a lot of data on that. We kind of know which ones are junk and which ones aren't and code for something, but I don't know what they do code for.
I guess
Yeah.
eventually that will.
Yeah, eventually, yeah. Yeah.
Along the same lines, is there a protein sequencing between the two?
What you do want to sequence a protein coding gene in order to find out like what genus or what family the mushroom's in. As I said, actually, yeah, let me show you a tree. Let me show you this. Here's a tree of all the mushrooms in the Gliophylales, where Gliophilum ciparium, which we have out there, is. And here's all the ITS sequences, and we made a tree out of it. And I color-coded it to show like each genus. Now, the ITS gene is really good at telling you if something is the same species or not. These 3 are one species, Neolentinus adherens, and these up top are Neolentinus kaufmannii, and they're practically the same. The difference is so slight. It's just this tiny little branch. But ITS shows you every little difference that there might be in the gene sequence to help you be able to separate them. But there are so many differences between this mushroom and a mushroom in a different family. There might be hundreds and hundreds of differences. It cannot figure out how to make the structure of the tree. It cannot figure out what family it's in. So you want to take a protein encoding gene that mutates slower and sequence that, and that might be able to tell you what family it's in or what genus it's in more reliably, but you'll never know if it's the same species as another species. You only know it's in that genus somewhere. So you want to do a combination of genes to be able to build the whole picture.
And looking at it from a different point of view, so this mushroom has a particular protein sequence that makes this
Yeah.
chemical. Okay, now I want to know what other mushrooms make that chemical.
Yes. Well, yeah, you could, if you did know what gene did that and what values produced that protein, you could blast against all of GenBank and find things that match that. But that could still be difficult because I would imagine that it's unique enough that you could find it. I've never tried it, but certainly such a thing is possible. So what's interesting about this is the genus Neolentinus shows in 2 places of the tree. It shows that perhaps really it should be split into 2 different genera. But it's lying, because here's the percentage likelihood that the tree is correct. There's a 100% chance that these are sister species. Great, but there's only a 24% chance that these belong together, that Gloeophyllum is more closely related to these Neolentinus than the Neolentinus down here. And there's only an 8% chance that this whole branch of the tree is correct in excluding this here. So you really can't tell with just ITS. Even the structure of the genus isn't obvious. I'm able to tell that, okay, these are 2 different species, these 2 are different species, but as far as what genus they really belong in, then the percentage likelihood starts to really drop rapidly. And if I sequenced other genes, you would get a tree that had much stronger, higher numbers there that you could believe. But this tree is basically guaranteed to be wrong. And in anything except showing you the most closely related species and whether or not they might be different or the same thing.
Danny, has there
Yeah.
been any, going back to Mycomatch, has there been anybody trying to replicate what you guys have done for different areas of the country?
No, we would love somebody to, because we've made the program so that you can add your own mushrooms and create your own area. You can go, here, add a new species, and then you can type the order of the family it's in and enter all the Alberta mushrooms that don't occur in British Columbia. If you had a list, you could add them in and you could make an Alberta version. But it's a lot of work and no one's volunteered yet. Yeah. But we have made that as easy as possible so that if somebody wants to do that, the program allows you to make your own whole set of mushrooms in there. Yeah.
If 10 people want to do one at a time or whatever, that doesn't.
Yeah, that would be great. Yeah. Yeah. So if you're interested, let me know and I can tell you more about how you might possibly do that. Make an Alberta version.
That would be a really interesting project
Yeah.
for citizen scientists.
Yeah.
to do.
Yeah.
You know, people, and not one person, but a bunch of people.
Yeah.
And have to have a bit of a.
What's remarkable is that this mostly represents one person, Ian, who did all the data entry for this. Yeah. That's crazy. He's been at his laptop since 1999. He's probably still there right now.
Probably not the same that.
Yeah, probably not. Oh, hopefully he's upgraded. Yeah.
And one of the things that isn't unique about him is that he's an insomniac. Any other insomniacs in the room?
Yeah.
You know, here's a good way to be.
It's a great idea. Yeah. 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 Yeah.
What about a project for a grad student?
Yeah, and that's
Or, yeah,
something we should
yeah.
really consider. And it's
Yeah.
very interesting when you're talking about that, because, or some of the things that you were mentioning about stuff that's missing in the literature. And I've gone through lots of literature trying to compare different things like Lexinum. And it's amazing what different characteristics everybody's looking at.
Yeah.
And you're, like, oh, so you don't have,
Yeah.
these guys don't have a spore color, these
Yeah.
guys don't have a this,
Yeah.
that guy doesn't have that.
But
and
you can
of these
all
things.
get to
Yeah,
Lexinum,
and
though,
so you can never
in different
get all
ways,
the
yeah.
characteristics, and it
Yeah.
starts getting frustrating.
Does this one also have the different names as they change?
Yes, it will put, let's see if we have, there. So for instance, Cystodermella cinnabarina was also called Cystoderma cinnabarinum, was also once called Lepiota cinnabarina.
So it'll
But-
pop up even if you search up one of those names, or I guess you couldn't get there unless you.
Ye- ye-
And we talked a little bit about this in my class the other day, is this, because I've seen people arguing about, no, it's this mushroom, no, it's that mushroom, and it's exactly
The
the same
same mushroom,
mushroom
yeah,
they're arguing
yeah.
about, they just.
Have
it's
different
just
names
like
for it,
have a
yeah.
different name. So the first thing to do when you're arguing is go and look and see if actually
Yeah.
the names are the same, or the names are the same for the same mushrooms.
There, you can do a list by alternate names, and now all of the possible names show up here. So, yeah, so you can basically find, see, Cinnabarina. Let's see here, Cinnabarina. There, Armillaria cinnabarina. You can call it that, and it'll pull up Cystodermella cinnabarina. Yeah, or Lepiota cinnabarina, same thing. So it tries to work with all the synonyms too.
Going back to this, you know, Rick has been working his ass off for years photographing.
Not really.
You know, and if we do have volunteers, we do have huge resources for Alberta mushrooms that we haven't used and aren't utilizing to the extent that we have.
One thing I should caution you of is when you do a blast, it gives you a button to make a tree, and it can make a little tree for you of everything that it found. So let's make this tree and download it where I can see it well. But it is really bad at it. You can't trust it's here. Let's grow it. It's see somewhat of the structure of the tree, but it's really, really, really bad quality for reasons I won't go into here. But there are free programs that make really good trees that you can get and do yourself. So BLAST is really brilliant for comparing DNA against other DNA and showing you the differences. But you wouldn't want to use it to make a tree.
So when you're looking at sequences, do you go straight to BLAST?
Yeah. Yes.
So, because you talk about UNITE as well.
No, UNITE only can give you sequences. You can give it a name, and you can find sequences that people have called that name. You can find out where they were, actually find on a map where they were, and then it can show you the sequence. And then you give that sequence to BLAST to compare it to something. Any other questions?
Would you use like Python and like matplotlib or something like that to make the tree?
Mate, the math behind it is complicated enough that I would never try to do that. I'd leave it to the program to do that. You could theoretically, but yeah, but it's beyond my pay scale to understand how to do that.
Why did you decide to tackle Cortinarius and Russula?
Pity? No, nobody else was? No, there's a good answer, really. For Russula, the founder of PSMS, Ben Wu, his specialty in the Key Council was Russula, and he devoted his life over 40 years or so. He had 1,000 collections of Russula that he had great notes on, and he dried and saved in his personal herbarium, and then donated them to UW. And a friend of mine, Anna Batsikalupo, did her master's thesis on those sequences. So by sequencing them all, got all 1,000 collections and sequenced them all and made a big tree of what they were. So there was now enough data to be able to learn something about Russula, and that was exciting. It was like, normally it would be, you would just pick away at it, a year at a time, working really hard, and maybe you would like learn one little thing, but with this much data, 1,000 local Russulas that showed most of the species that were out there, to have their sequences and be able to compare them and put names on them and start naming the ones that weren't, that was like exciting that progress could be made in a really difficult genus. And that was like a stepping stone to just opening the doors to knowing so much more than we used to. And that was pretty exciting. And Cortinarius, just because it's the biggest challenge. And because I was curious how many there are. So the Cortinarius is the world's largest guild genus. And since it's such a big project, I'm only doing step one right now, which is just collect DNA and see how many different species we have. Don't try to figure out how to key them out or anything, just like count them is the first thing. And I'm at 524 for just like BC and Washington and Oregon. We're about 524 species in that genus. So we've got a lot of data. Yeah.
So Danny, earlier you said like for some of the sequences that don't have names, like it comes back with a PNO2 or whatever.
Yeah.
in future, will those be turned into names or?
Yes, that's going to be a slow process because describing them, you need to have all the microscopic data and all the, it's like, sort of like a few a year are being described and maybe 10 a week are being found. So there's no way that can keep up. So
Scott Otsuni is doing a group to get people together to start describing
Yeah.
species
Yeah, and-
methodically. So hopefully the numbers of people
Yes,
doing that
yeah.
will increase.
And we're also going to just like give them at least provisional names. So even though we don't have a description, we'll at least give it a name, and so we can start talking about it with a name, and then let people catch up. And yeah.
And we've actually had some workshops at GAMFs on describing mushrooms from people like Dr. Jim Gins and
Oh, yeah.
whatnot, and they've been absolutely wonderful. And it's a lot of times it's hard to do it yourself, but if you've got a group of 3
Yeah.
or 4, and say like, what is this color?
Yeah. Like,
you said,
Alfredo did most of the work, but there's like 20 of us that contributed to the paper that named a bunch of the new Pluteus that he's going to talk about tonight. We did, very little, but I mean, each person does like a little bit to help, and then you can accomplish a lot.
Yeah, so we've done some of that stuff, but that's one of the things that we should be doing on a more regular basis. I mean, even talking about what the smell of a mushroom
Yeah.
is, because subjective, and everybody's smell of a mushroom is
Yeah.
valid because they're all different. And, what is this color? What is this? And get a consensus.
Yeah.
Yeah, it's interesting.
Yeah. Yeah.
I wonder if you could demonstrate something?
Mm-hmm.
You have a list with Agaricus, and then the list of Agaricus nensis. So if I wanted to know what similarity Agaricus mushrooms to that one, because that one I believe grows in Brazil or southern Florida, is there's any in Alberta that grow that are similar to that?
So you can start with, let's see, Agaricus augustus here, and let's go list similar species. So here is Agaricus augustus at the top, and here's all the other mushrooms in the world, and what mathematical percentage it considers them to be identical by just counting the number of characters they have and how many match and how many don't. So you can kind of go through that in backwards order and play with it that way. So it's got some really cool features, the way it the way it can present the data to you, and it can also do things like, let's say, nobody, like, a lot of people use it to just basically click the boxes and see what it has, but not a lot of people know about these other cool features that it has. So for instance, I can say it's a red mushroom, and I've got 142 possibilities. You can say something like, where is it? List. Match best characters. Okay. So this tells you, if you told me that it had a dry stem, 95% of the mushrooms that are red also have a dry stem. You're not going to get very far. So don't bother looking at that. But way up here, if you can tell me that it has a thick ring veil or that it smells like almond, there's only 1% of the mushrooms, or one mushroom has that. So why don't you try to look for these things? And if you can find one of these things, I'm going to be able to tell you what it is. But if you're going to tell me that the stem's dry, you're not going to get anywhere. So it can help lead you to the next step. Like concentric scales on a red cap mushroom. It's only one thing. Like, you don't need to even know about the warts. If you see a red mushroom that has concentric scales at the bottom, you know it's Amanita muscaria. You just click on that one. So I'll do that again to show. So here's the red mushrooms, and here's all the best characters. So I can look through this and I can say, so if any of these at the top, any of the first few pages are true about your mushroom, it's going to be able to tell you exactly what it is. So you can scan through and say, oh, it's got concentric scales. Great. Click on that and say, I'm going to tell you what it is. There it is, Amanita muscaria. And there's the There's the picture. Group, yeah. There's the picture with the concentric scales.
So
Yeah.
that raises a question. Well, something we call Amanita muscaria here isn't red at all. It's orangey yellow or yellow with the scabers on it.
Yeah, our local species can be red, it can be orange, it can be yellow, or it can be white. All the genes that have been sequenced are basically identical. It's they're just color forms of each other. But it's not the European Amanita muscaria. We have these kind of yellowish tinged veil. It's not pure white. The warts often have a yellow tinge to them. And the true Amanita muscaria is got pure white warts and grows under different trees. And it does occur here, but only like 5% of the ones that I've sequenced turn out to be Amanita muscaria. The rest turn out to be our local species.
And so have we settled on a name for the local?
Oh, I've looked at that for so long. I can give an hour and a half talk on what do we call our local one, and it goes into all the rules of nomenclature and why there's no good answer. We have a variety name for it, variety flavivulvata, but it's too different to be a variety of muscaria. It needs to be its own species, so we can't use that. There may be a name, chrysoblema, because Peck described Amanita chrysoblema as a white East Coast mushroom, and that may be the same as ours. So we may have a name, but now we can't prove that that's the one he found. There's 2 or 3 things that it could be, but I've tried to sleuth out and find logical reasons why it couldn't be anything else. So I believe that Amanita chrysoblema is what we should be calling our Amanita muscaria, but I just can't prove it yet.
Do you think it's the same for Alberta?
Yeah.
I've read multiple sources that they're now considering chrysoblema the new North American species, and a lot of people are saying, like a lot of sources are saying that.
It's catching on. All right. Somebody's believing me. Yeah. Yeah.
The bigger identification groups and a lot of like the experts that are on there are saying that's the new name now.
I'm good.
But
That's a
not, like,
catching on. Yeah.
It's not fully been accepted by the
No.
community.
Yeah. No. It was just me posting like I think it's this, and then some people believed me and some people didn't, and then I said well, and then I like gave more sort of persuasive arguments, and more people believe me, but still some people insisted it wasn't, and until somebody just goes and like publishes and makes it, we're just going to be arguing about it. But yeah. Yeah. Yeah, I've looked at other possibilities, and they don't seem as likely. You can read all about it if you go to my DNA website. It's alpental.com/PSMS/DDD for Danny's DNA Discoveries. And go to the Amanita page. Amanita section, Amanita. And here we go. So this is a few pages that go through all the possible names. So here's all of the things that we know about the genetics. Here are all the different clades. Here are all the possible names that I found that it could be, and I chose chrysoblema, and here's the list of reasons why I might be wrong. So you can read all of this and decide for yourself.
And what about the Siberian version? Was there a difference between what the shaman of Siberia?
I'm not sure what the distribution is. I'm not sure which one would have been in Siberia. Yeah, I have to look at them. Let's find out. Let's go to Unite that has Amanita muscaria. Where is it? 95% of them turn out to be the same thing, so you have to get lucky. Let's see how many different mushrooms are being called Amanita muscaria around the world here. Okay. 3 major ones, 127. The internet just got slow, which is a real bummer because I'm really eager to get the answer to this. Let's see, and this one, 393 sequences are this species and 29 are this. Let's take a look. What is it? The internet has been so fabulous until now. Maybe the smoke is interfering with the Wi-Fi. Oh, unfortunately, it's really... Oh, there we go. Okay, so this species is our West Coast species, and it's also found in Japan. How about that? Yeah. Whoop. Down. So this is not the real thing. This might be the one that's ours. Let's see what we got here. Hasn't. Oh, here we go. Here's one that's all over and a little bit in Japan and also in Europe, but nothing in Siberia yet. This is our last chance here. Let's close down the ones that are thinking too hard. Ah, here's something on the Siberian Peninsula here. Let's see which one this is. All right, so this is the species that occurs in Siberia. I looked at the maps of the 3 different species, right? I found one that is pretty close to Siberia. I'm going to take a sequence of what it is, and I'm going to blast it against my reliable database of Amanita sequences whose names I trust. And let's find out which one is in Siberia.
Is your database available anywhere?
Yes, it's at the top of my, at the top. Right, it's at download all my FASTA DNA data, my trees right there. It's the very, very top link. Okay, survey says Muscaria Pacific Northwest 03. Wow. No, wait, Muscaria 9983. The highest match, it's the real Muscaria. So, so Siberia and Europe have the real Muscaria. Yeah.
Interesting.
Okay, any other questions? Yeah, if you come up with some afterwards, feel free to find me and pick my brain. Yes?
Just that you showed us where we could download your things, but I didn't see the web address at the top of the scanning.
Oh, that's a local copy. See, there we go. There we go. alpental.com/PSMS/DDD. Yeah, thank you everybody. Appreciate it, yeah.